The Central Dogma of Biology by Caden.
Sienna.
Camden.
Let's talk about DNA and its structure. Deoxyribonucleic acid is an acid that contains the genetic instructions for the development and function of all living things. The main function of DNA is the long-term storage of information. Without DNA would mean that your cells wouldn't be able to regenerate all the proteins lost to normal turnover, such as growing a new finger after it's been cut off. DNA is made up of molecules called nucleotides. A nucleotide contains a phosphate group, a sugar group, and a nitrogen base. The first part of a nucleotide I'll talk about is the phosphate group. Its purpose is to store energy in order for polymerase to function, which we will cover soon. The second part of a nucleotide is a 5-carbon sugar, or deoxyribose, and is a key building block to the DNA structure. Its carbon-based structure allows for a stable and strong backbone for the DNA. The last element of a nucleotide is the nitrogenous bases thymine, guanine, adenine, and cytosine. The nitrogenous bases are composed of carbon and nitrogen rings that form hydrogen bonds between opposite DNA strands to form the twisted ladder shape of DNA, or the double helix. On the double helix, the two strands are anti-parallel, meaning the strands run in opposite directions in a 5' to 3' direction. 5' to 3' means that the phosphate group of a nucleotide is bonded to the third carbon of one deoxyribose and the 5 carbon of another. This was concluded by two scientists in the 50s named Watson and Crick. These scientists also found that the backbone of DNA is made up of chains of deoxyribose sugar and are covalently bonded to phosphate groups. A covalent bond is appropriate for the backbone because it is very strong and can hold the structure together. On the other hand, the nitrogenous bases are composed of hydrogen bonds. These bonds are weaker than a covalent bond and can be easily disrupted. Think about a wave crashing on a beach. The sand is loose and can be easily disrupted. This allows the DNA strands to separate for transcription, which is the process of copying DNA to RNA, and also replication, the process of copying DNA to DNA.
DNA replication is the process by which DNA makes a copy of itself during cell division. This process contains 9 different enzymes. The first is DNA helicase, which breaks the hydrogen bonds between the bases while unzipping the double helix. In order to do this, the helicase enzymes move in both directions to form a replication bubble. At the end of the replication bubble, there are replication forks where new strands of DNA are extended. Secondly is gyrase. This enzyme is before the replication fork that unwinds the supercoiled DNA. In order to keep the DNA strands apart, single-stranded binding proteins hold the DNA apart to prevent it from coiling back together as well as stabilizing the DNA. Next enzyme is primase. This enzyme adds RNA nucleotides, which acts as a primer for DNA nucleotides. These primers are around 10 RNA nucleotides long. To add another nucleotide, you must have a primer because DNA polymerase can only add nucleotides with a primer. Next is DNA polymerase III, which adds DNA nucleotides to the RNA primer sequence in a 5' to 3' direction. Since the two sides of DNA are anti-parallel, one side goes in the 3' to 5' direction, and the other goes into the 5' to 3' direction. The DNA polymerase III is only able to add nucleotides to the 3' end of the DNA strand. There are 2 strands in DNA polymerase III. The leading strand synthesizes a complementary strand on one side of the template in the 5' to 3' direction. The lagging strand moves away from the replication fork where Okazaki fragments make short strands of DNA. After, DNA polymerase I removes the RNA primers and replaces it with DNA nucleotides. Then, DNA ligase joins the Okazaki fragments to form a single DNA strand. Finally, DNA polymerase I proofreads and repairs mismatched bases and removes any abnormal bases. One type of nucleotide is a nucleoside triphosphate, which is a molecule that contains a nucleoside bound to 3 phosphate groups. The difference between RNA and DNA is that RNA is single-stranded and contains a ribose as a sugar, but DNA is double-stranded and contains a deoxyribose. Also, DNA contains thymine, while RNA contains uracil.
Transcription is the process of RNA copying a strand of DNA through the phases of initiation, elongation, and termination. For prokaryotes, this occurs in the cytoplasm, and for eukaryotes in the nucleus, mitochondria, and chloroplast. Initiation starts when RNA polymerase II attaches to the promoter on the DNA strand. A promoter is a region of DNA that is located near the transcription start point. Then it creates an RNA transcript in the transcription bubble. Transcription factors then stick onto the promoter, binding RNA polymerase II to the DNA. Then, in elongation, it starts moving in a 5' to 3' direction. RNA polymerase II continues to synthesize an RNA transcript. While doing this, it creates the strand anti-parallel to the template DNA. Energy is provided from triphosphates. Finally, the RNA polymerase hits a transcription terminator which releases the growing RNA strand and breaks off the polymerase. Once the RNA for a certain section is completed, the double helix quickly reforms, displacing the growing RNA strand. Multiple RNA molecules can be transcribed from the same gene simultaneously. RNA modification is the process of a newly transcribed pre-mRNA that changes to the mature product of mRNA, which only happens in eukaryotes. While non-protein coding regions of the RNA strand are removed, exons, which is the real gene and are expressed in the coding sequence, while introns are non-coding in between the sequence. The introns are then spliced out of the RNA and join the exons. The two RNA splicing enzymes are SRNPs, which are small nuclear RNA, and proteins, and the other is the spliceosome, which contains several snRNPs and recognize the splice sequences. During RNA processing, a 5' GDP cap is added to protect the ends of the molecule by adding a cap consisting of modified GTP, as well as adding a 3' poly A tail, which is a longer tail where mRNA lasts longer and produces more protein. The 5' GTP cap and 3' poly A tail protects the mRNA from enzymes in the cytoplasm that attacks the mRNA.
During translation, RNA molecules are used to code protein assembly at the ribosome. In initiation, the small ribosomal subunit binds to the mRNA with the start codon. The small subunit reads the RNA, and a codon is 3 nucleotides, which forms 1 unit of genetic code. Then, the tRNA with the complementary anticodon bonds to the mRNA small subunit complex. An anticodon is 3 nucleotides forming a unit of genetic code in a transfer of RNA molecule which corresponds to the complementary codon. The large ribosomal subunit then joins amino acids to form a polypeptide chain. Next step is elongation. The mRNA codon is now in the A site. The tRNA with the complementary anticodon hydrogen bonds to the codon in the A site. Also, the tRNA brings the appropriate amino acid to the ribosome. A peptide bond formation takes place to connect the amino acid in the A site with the amino acid in the P site. The mRNA molecule is read and the new amino acids are added in the 5' to 3' direction. During translocation, the ribosome moves towards the 3' end of the mRNA strand, and because of the move to the ribosome, there is a shift in the tRNA binding site. tRNA then leaves the ribosome from the E site. Next is termination, where the adding of amino acids to the polypeptide chain continues until there is a stop codon on the RNA. A release factor binds to the stop codon and hydrolyzes the completed polypeptide from the tRNA, which releases the polypeptide from the ribosome. The ribosome dissociates into the 2 subunits, and the amino acid chain can now fold into its secondary, tertiary, and quaternary structure to become a functional protein. Proteins have several layers of structure, each of which is important in the process of protein folding, which is where a protein gets its shape.
Today, you learned about DNA structures, DNA replication, RNA modification, transcription, and translation.